Okay, this is a talk about macroinvertebrate and water quality, and it's related to the University of Rhode Island here. Leon and I worked on this project, and this is a report on it. We also can call it the health of the pond at the in terms of pollution level, and I call it a quantitative analysis. It's more or less a semi-quantitative, but for people who were here a few years ago, there were no paved parking at the bottom of the hill. So in recent years, they put in these big paving parking lots, huge ones, and the big question is whether or not those affected the water quality in the pond, which also would affect the water quality in the big wells that we have at the bottom of the hill. We get our water at URI from the wells, and the wells then pump the water up to that big blue monster at the top of the hill, and then that circulates the water throughout the campus. And so we're interested in doing that. One of the ways to do it is to collect macroinvertebrate from the pond, identify the different macroinvertebrate, and then determine the quality of the pond based on what we found in terms of those macroinvertebrate. So the 30-acre pond is down the hill behind the turf farm, and it's about 15 acres. It's a freshwater lake. Why they call it 30-acre pond when it's only 15 acres? Maybe back 1,000 years it used to be 30 acres. What is a macroinvertebrate? They're organisms that do not have a backbone, invertebrate. They're visible without a microscope with the naked eye. They require oxygen. Some come from the air and some comes from dissolved oxygen. And the organisms are available to plants and to other animals, so they get eaten up by larger animals such as fish. Why macroinvertebrate? Because they are sensitive to changes in the ecosystem. In other words, they're sensitive to some pollution aspects in the pond. Many live in an aquatic ecosystem for over a year. They cannot easily escape changes in water quality, so they'll die out if the water becomes polluted. They can be collected very easily from most aquatic systems with inexpensive equipment, and I'll show you some of that equipment in a minute. They diversify, their diversity determines whether the ecosystem can support fish, birds, and other wildlife that eat them. Where do they live? They live on the water surface and the bottom of ponds, so it's an ideal situation at the bottom of the hill to use this technique. They're under grasses and undercut in terms of streams. They live at the river bottom, under and among the rocks, and on or among vegetation in the water. They like water. Some of them like water tumbling over rocks, and some like smooth water. So pools of slow-moving water or a pond is an ideal situation. Okay, it looks better up there, but these are some of the macroinvertebrate that we can find down in the pond. If anybody wants to come and help me in July, you're welcome to do it. This is the cycle. Look at that, 18 seconds so far. Macroinvertebrate, this is the food web actually. You get some light going in here, and then you get plants and so forth. You get some dissolved organic matter, you get microatoms, you get flocculation, and then you can come up with shredders, you can come up with collectors and predators and scrapers. Those are some of the categories. I don't think we get mussels here, except, yes, we did get a couple mussels. Here's the procedure. Get out in that water. You can see the sampling device. There's one that's just a filter here. There's another person putting it directly into a bucket. And you notice there's waders on these people. There's boots or waders, depending upon which ones they use. This one has a wader. This one must have boots. Oh, no, this has a waiter. It's just harder to see. So you have to put these things on so you don't get your feet wet. There's a nice collector. It's just a another screen material here. And you'll notice this is 2009 one we did it then in 2011. We did 3 feet out from the shore and 8 to 10 feet out from the shore. We took 2 sets of samples. They match pretty well because the pond doesn't get too deep. And then we bring them back to the lab and we count them and we identify them and we make a list of all of them. You see my bucket? There's another one of the tools you need. The bucket. And so here's identifications here, and they're identifying these. See how interested those students are in making sure they get it right. And that's another important thing. You don't need a microscope to see these things, although we did have a few magnifying glasses so they could be identified. Here's another method. Another group. We had half a dozen groups doing this, and they're looking at them in a plastic dish. They've gotten them out of the buckets, putting them in plastic dishes, and now identify them. So there's 3 major types of macroinvertebrate. One is pollution intolerant. That means they'll die if it becomes polluted. And the second one is somewhat pollution tolerant. And the third one is pollution tolerant. So if your pond's becoming polluted, the pollution tolerant ones tend to dominate. My muscles here. Okay, so here's 3 categories of this system. Sensitive to pollution. That's what we want to identify, those kind of things. Look at that, water pennies even. And these, we also want to identify these. If this grows in terms of numbers, then we're in trouble. If this grows in terms of numbers, then we have a pond that's really in good shape. Here's some more examples. We have leeches, water spiders, water scavengers. Oh, what fun to collect all these things. More worms here, a whole bunch of worms, and the you can see a clam-like and mussel-like macroinvertebrate. What equipment do we need? We need screens, we need trays and petri dishes, magnifying glasses. We didn't really dissect any of them. Maybe a pipette, small paint brushes maybe. Identification charts are really important. And then a biotic index charts to tell us which ones are pollution tolerant, pollution intolerant, and intermediate. So we take a trip to 30-acre pond. It's a nice walk on a nice summer day. Collect macroinvertebrate near and far from the shore in buckets. We then carry the buckets back up here. Use identification charts to identify 100 different ones, the more common. Collect more samples as needed. Add up the numbers of macroinvertebrate. It's helpful to search over a fixed area so you know what that square footage was, and you collect in the same square footage the next time around. And you determine the density then, how many macroinvertebrate there are per cubic foot. We know the depth of the water as well. And or we could just do it per square foot, which is both are reasonable for the pond there. We'd like to collect over different seasons. If we collected in the fall, we might get different answers, and maybe Dr. Thiem is going to try to collect some. He's got all the waders, he's got the buckets, he's got the screens. You just need to go for it. We are worried about temperature changes from year to year or season to season, dissolved oxygen, algae growth, because they affect the quantity and type. And we want to determine trends. So is the pond becoming more polluted, less polluted? We couple the collection with water quality measurements, especially temperature, pH, and some other material, turbidity. Okay, so then we classify. A is pollution intolerant, we call class 1. B is somewhat pollution tolerant. C is pollution tolerant. Class 3, 30-acre pond, the protocol. Several samples and individual collect at 3 feet from shore and roughly 8 to 10 feet. It was hard for the students to determine whether it was 8 feet or 10 feet, so we have to have a range of feet there. And we evaluate the entire collection, a fixed number, and proportions. Of the 3 tolerant, these are the discussions we have of the 3 tolerant levels, what had the most, the least, how many species were identified, what is the health of the pond, and you can find a lot of the description of the macroinvertebrate on the web. So in 2009, this is what we got in the categories. A is the good category, and B is the intermediate, and C is the bad. So you can see we found stoneflies, mayflies, caddisflies, dragonflies, a whole bunch of different possibilities there, even more possibilities. And you notice we had 10 freshwater prawns in 2009, and the next biggest numbers were the damselfly. Okay, so then this is a comparative method and used by myself and Dr. Thiem. You multiply the numbers of each category by its quality factor, and I set the quality factor. Find the total for each category, divide by the total number of species, get pollution indices, and this is a comparative method. Hit the wrong slide. Okay, and so this is the data in 2009. A total of 65 macroinvertebrate in the small square footage that we surveyed. And the quality factor for intolerant, it's a 3 number, and for tolerant, it's a 1. 24 tolerant, 18 intolerant. That was in 2009. There's the equations that we used. Quality factor times the fraction. And this is our results in 2009. 0.83 was a reasonably good number, so we could pretty much say that the pond wasn't really polluted in 2009. Try to keep that number in mind, 0.83. And then in 2011, we went and found a lot of caddisflies there, and a whole bunch of riffle beetles, and a number of black flies, and a number of stoneflies, a little bit different species, and some nuns. And then there was 65 that year in pretty much the same area, 69 in 2009. Again, the intolerant were 46, pollution tolerant 24. The number of intolerant went up, which is a good sign. And so again, the same equations that we used just earlier to calculate that. And now we have a pollution index for the intolerant ones of 1.7, almost double the 2009 level. And that's the key number to say whether the pond is healthy or not. So we can pretty much assume that between July of 2009 and July of 2011, the health of the pond improved. Here's a comparison for the 2 years, 3 and 1.71, the key indices in this. And the question is, why did the pond improve? So you want to look at some of the data that we took. Dissolved oxygen was about the same, the temperature was the same, the time of year was about the same, the pH was about the same in both years. Even the rainfall was about the same in the 2 years. 2009, 7.63 inches, 2011, 7.32 inches. So is it true that the parking lots have holding ponds or some way to filter out what's in there?
The ones closest are porous, well, were porous pavement at one time, but right now probably mostly sheet flow off of them. So no, there is no holding pond.
So it doesn't seem that they're running off into the pond, which is a good thing. since we have lots of cars there sometimes, especially when Ryan Center is operating. And so I'm not sure what caused the improvement. I just couldn't find a thing, but we can conclude that the health of the pond has increased, which is a good thing, because the pond is right next to the 4 wells that we use for drinking water here. Okay, so thanks to Rhode Island Water Resources, NSF, and the LSAM program, Times2 Academy, and the Providence Academy for International Study. Times Square and Providence Academy for International Studies were the students involved in these experiments. And we're almost ready to go home here. What does that say? Total time for the slideshow, 14 minutes, okay, pretty good. Are we out of it? Okay, good. Okay, anybody have questions? Okay, that can, yeah.
So, did you test for like petroleum type of compounds at all? I mean, you said there's some
No,
concern.
no, we didn't. Dissolved oxygen, pH, nitrogen, temperature. Some miscellaneous chemicals. Okay, other questions? Did you know about the, I meant to ask you this if we could ask you during your talk, but since we got 2 minutes left, I'll ask you a question. You heard about the Johnston dump? The smell is really bad up there. What happens is they have these pipes going through the solid mountains that they have, and the gas collects in there. They feed it to a place where they clean up the gas, and then they generate electricity.
Yeah.
When Irene came along, they flooded the landfill. All those pipes are filled up with runoff from the landfill, and so it's kind of growing organisms and bacteria in there, and the smell is really terrible. Can we take these generators and loan them to the dump to do something with that water?
I've talked to the powers that be.
You what?
I will talk to the powers that be
Okay.
as far as the opportunity there.
I think they're pulling their heads out and they're willing to do something
Yeah.
at this point. Okay, good. Okay, thanks for coming, everyone. Appreciate it.